Welcome everyone. Good morning from Vienna. This is 11 o'clock in Vienna. Wherever you are, we wish you all a superb day because we're going to have a wonderful 1 hour now. In the next 1 hour. Welcome to our side event. This is a side event at the World Food Forum, and the event is brought to you by the FAO and the IAEA, the Food and Agricultural Organization of the United Nations, and the International Atomic Energy Agency. And what do we have for you in the next 1 hour? We have some interesting lab stuff coming your way and the lab experts joining us here for the chat. So please monitor the chat, pose your questions as we go along. We don't have much time, it's only an hour. My name is Ayhan Evrensen. I'm working here at the IAEA in Vienna as a communication expert, and I'm joined by my colleagues Sinead Harvey and Eleonora Kolzani, and later I will also introduce you to our lab experts. We will talk to you about plant breeding and sterile insect technique. How do they affect our life? How do they help us tackle the world hunger? And how do they really help the climate change? So there are so many interesting aspects that we will talk about. And the World Food Forum is a good opportunity for us to bring these into limelight. How on Earth nuclear technologies are used in these topics. So for this, to bring you closer to the science, we went last week to our laboratories just outside Vienna, about 35 minutes outside Vienna. We have a laboratory complex in Seibersdorf, and the Food and Agricultural Organization, FAO, and the IAEA run these labs together. We work on very different scientific areas, but we will highlight two of them only to you today, and you will see those in the video that comes up very soon. We have two short sessions of 7 minutes for each lab. One is about tsetse flies, one is about plant breeding, and how we use nuclear science in bringing us closer to the Sustainable Development Goals. So let's take it now for the video. Stay with us and don't forget, pose your questions in the chat. Later we will come back together.
Good morning. Welcome to the FAO IAEA Insect Pest Control Laboratory. My name is Mark Vrezen, and I'm the head of this laboratory. Maybe you're not aware, but every year this world is spreading insecticides over the planet worth 60 to 80 billion dollars. That is a lot of insecticides, and this is obviously very, very bad for the environment. It's very bad for the farmer who has to do the spraying, and it is very, very bad for the consumer, because there will always be residues at the crop. Therefore, there is a tremendous demand for insect pest control tactics that are not only friendly or neutral to the environment, but also very, very effective. And this is the technology that we are working on. It's called the sterile insect technique. Its principle is very simple. You bring the insect that you would like to control, you bring it into the laboratory, you rear it in very, very large numbers. You sterilize using ionizing radiation the male sex. You release those male insects in the target area where they will mate with the virgin females, and there will then be no offspring. So basically, it's a type of bird control of insects. This is the type of work that we are doing in this lab, and let me introduce you to Chantal de Beer, who is the head of the livestock pest group, and she will explain to you some of the experimental work that we are doing here.
Very welcome at our field cage mating experiment. Let's go inside and then I will show you how the experiment is done. Now we are inside our experiment. So this is a field cage. It's a semi-field cage, and I have already released the females in here. So if you look around, you will be able to see the female tsetse flies inside the cage. So we release them inside the cage a little bit before the males so that they can be happy and be ready for the males to be received. So we have the females in here, And now we will be releasing our males. We have 3 types of males. I've marked them yellow, red, and blue. The yellow males are males that have been irradiated with gamma rays. The red males are the males that have been irradiated with X-rays, and the blue males have had no irradiation. The purpose of the experiment is to see how these males compete for the females inside the cage. So let's open them up and see what they do. So we will open the cages like this so that they can go outside. So we will give them a little shake and they will go outside. So this is a very interesting part of my work. I like being inside the field cage with the insects and seeing what they do. So we shake them and they will go all the way outside so you can see them around. Okay. So you can see them on the side of the cage. If you look closely, you can see some of them have blue dots on them, and some of them have yellow dots on them, and some of them have red dots on them. So now they need to find our females. So they are all here and around here. So we have to wait a little bit so that they can find our females. At the moment, when we look around, I don't see any mating pairs at the moment. So we just have to give them a while to find the females. So while we're waiting for them to mate, let me tell you a little bit about why they are a pest. Tsetse flies are only located in Africa, so it's an African species. They are vectors of a small parasite that you can find in cattle blood or human blood, and they are a pest. They will give this disease to the animals, and this will be very bad for the farmer because the animals will get sick and they won't be able to produce lots of milk. They will also not be nice and fat to be eaten. And this is why we try to control these insects. The life cycle is very unique, like I will show you later. So SIT is a very nice thing to be using them as birth control because one female will only produce 6 offspring in her lifetime. So let's go and see if we can find a mating pair. So here we have our first mating pair. You can see the male is on top of the female. The male is from the yellow group. And the male will find the female, they will attach, they will start mating, and this mating process can take 3 hours. Afterwards, the male will give sperm to the female will store the sperm inside her body, where she will then fertilize her offspring. So now let's collect the pair. So we will collect the pair and we will keep it. So we will take the time of when they started mating, and we will also record the time that they took to mate. So we will collect it in our specialized collecting tube, and then it will go inside a little tube like this. So we put it inside like this, and then we will keep it and record the time, how long they take to mate. So normally the experiment will take 3 hours. So we'll put the females in, and then these 3 test males. We will be inside the cage for the entire time frame. We will record it for 3 hours. All the pairs we will collect, and then we will record how many of one group, how many of the yellows mated, how many of the red mated, and how many of the blue mates mated. We will also record, because we have 45 females in here, we will also record how many of these females mated. So we want them all to mate. Then at the end of the experiment, we can determine the radiation that we gave them. You know, remember, we gave them gamma rays or X-rays. Which of the radiation affected the ability to mate the most, and which affected it the least. And then if we have this information, we know what type of radiation to give them if we're going to release them, when we release them in the field for the SIT campaign. So after we've done this experiment, we will catch all the flies back, and they will be included back into the colony. As we are now done in our experiment, let's go see the feeding process. Very welcome to my feeding room. This is where we feed our colony flies. They are fed on these artificial membrane systems, so that we don't have to use animals. The membrane is made of a silicone membrane, and we put the cow blood underneath. The blood is collected from cows, we shake it, and then it stays in this liquid form. So the flies are inside these cages. You can see them inside there. So they need to put their mouth through the cages, through the membrane, and suck up the blood with their straw-like mouth parts. We give them the opportunity to feed for 10 minutes. After that, they are nice and fat and full of blood. We will keep them then for us to produce for us offspring. I have an example of their offspring. They are very unique flies. They do not lay eggs, but the female will become pregnant with the larvae. This is the larvae, so you can see them wiggling around. Those are the ones that have just been born. They will become darker, and they will be black, and then they will be like this for a month, and then we will have new flies. Thank you so much for visiting us at the Stacey Colonies, and stick around for more questions later on about our work. Thank you.
Hello, welcome. We are here standing in the experimental field of the Joint FAO IAEA Centre for Nuclear Applications in Food and Agriculture. Our research works with the improvement of plants, all kinds of plant species. Right behind us is a crop called sorghum, which is also known as the poor man's crop in the developing nations of Asia and Africa. So our job with plant breeding, the science of plant breeding, is to be improving the genetic potential of the plant through a variety of mechanisms and enabling those plants to be performing better, to give higher yield, better quality of the produce, and also resilience against shocks, stresses, such as that comes from climate change. High heat temperatures would reduce the crop yield. Pests and diseases that come out of the blue, that can also reduce the yield of the crop. And that is a big hit to the farmer's produce. And in subsistence farming conditions, which is smallholder agriculture that prevails in the developing countries of the world, where the produce goes mainly to satisfy the hunger of the family. And it's the rest of it that goes into the market. So it caters to both food security, nutrition security, and to the income of the farmers. So to prevent the loss of a crop is significant. So the technologies that we use are normally to improve these characteristics of the plants. Traditionally, plant breeding takes a long period of time where the genetics of the plant or the DNA evolves over the time in order to improve over the evolutionary period. We do irradiation of the seeds of plants to change their genetics in such a way that we can select for better plants that can be cultivated by the farmers. And like I said, these better plants can be anywhere from higher yields to better resilience. And now the technologies that are coming in, big data, artificial intelligence, machine learning, genomics, DNA sequencing across the entire genome of an organism, all of this comes together to help us develop a new crop variety in a much shorter time that can then reach the farmers and they can cultivate it, and that can go towards the food security locally, nationally, regionally, and even towards export income for the countries. So this is the science of plant breeding, and the high-end technologies that are improving the science and on a day-by-day basis is something that my colleague Norman can talk to you about right now.
Hello, I'm Norman. I'm a geneticist, and I lead a team here at the Plant Breeding and Genetics Laboratory that mostly works on genomics. A plant is a product of its genome interacting with the environment it grows in. And it can only be as good as the underlying genes it carries. And plant breeding is a continuous process of recombining existing genetic variation into a new crop. In nature, in wild species, the process of evolution is constantly ongoing. There's random mutations filtered by the environment, and natural selection, as Charles Darwin has coined it, takes place all the time around us. And that's why all individuals of a species look fairly different. We have a different situation in our crops because millennia of domestication have unified the genomes, and the genetic variation in most of our crops is very small. That's where mutation breeding comes in, where we mimic the process of natural evolution with a higher rate of mutation. We irradiate plants, inflict DNA changes, and then it's not nature that filters, but the breeder and the scientist filtering those plants to find the better ones for the next cycle of breeding. To do this, we have to select the plants, and we do this by looking at the plants themselves. We would look at their appearance, their chemical composition, and there we have new tools like multispectral imaging and all sorts of complicated chemical assays to, in high throughput mode, screen through hundreds of thousands of plants. At the same time, we look at their genomes to find out what these DNA changes are that we actually inflicted, and then all of this big data together we analyze with modern computer science, such as artificial intelligence, machine learning, and other approaches to link the genotype with the phenotype that we see and want, the new trait that the farmer ultimately requires and that adopts our plants to the changing environment. So this is all sophisticated science and requires a multidisciplinary team of scientists, which we have here at the PBGL, and let's go and meet some of them.
Hi, I'm Susu. I'm an agricultural economist from Austria, and I'm also learning about plant breeding. Today, I'm extracting DNA from a coffee plant to do genomic analysis.
Hi, I'm Adel from Egypt. Today I'm preparing PCR for marker development. I'm chemist.
Hi, I'm Manuel Morales from Puerto Rico. I'm a robotics engineer, and here I do data analysis for plant breeding.
Hi, I'm Faith from Kenya. I'm a molecular biologist, currently working in vitro tissue culture and on subculturing banana.
Hi, I'm Samira from Iran. I studied plant breeding and genetics. Now I culture leaf disc to develop cell suspension culture.
Thank you for listening. Please stay on for the Q&A session that is following this.
Cool stuff, and we're back, and we're joined now with those 4 colleagues you have seen on our videos. Do we see them? Yes. So I'll start. We've seen on the sterile insect technique 2 colleagues, Mark Friesen from Belgium, who is the head of the insect pest control lab, and his favorite insect, guess what, is the tsetse. So he's an entomologist. He has 30 years in entomology. Welcome, Mark. Good morning.
Good morning.
He's joining us in Seibersdorf in our labs outside Vienna, as well as Chantal next to her, who joins us also from Seibersdorf. Chantal de Beer is from South Africa. She's a research entomologist. I'm guessing your favorite insect is also the tsetse. We've seen it very lively in the videos. You've joined us last year in March, but you have 15 years of entomology background. So welcome joining us as well. Thank you. And then we have our plant breeders, Shobha Shivashankar from India. She's our head of the plant breeding and genetics section. As I said, from India, she has agronomy and plant biology experience for over 25 years. She joins us from the building where I sit in Vienna, but her colleague, Norman Bartmann from Germany, is joining us somewhere in a green field. No, that is also Seibersdorf. He's working in our lab out there. Norman is from Germany. He's been working on plant development and genomics for over 20 years. He joined us, the FAO IAEA Joint Center, 3 years ago. So welcome all of you. We have a good number of attendees listening to us. We've seen the videos, and this is weird stuff you are involved in, really. Each time I go out there in your labs, I'm just amazed what you can do with radiation, with nuclear science, and with everything. But the underlying thing for me is how do all these things connect to nuclear science? So we need to get a bit deeper into this. Can I start with Shoba, please? You said that irradiation brings mutations. Yeah, you force mutations. Do you force mutations on the crops, or how do you do that? And related to that, what is the connection to climate change? How do I get this connection?
Thank you, Ayhan. So normally mutations happen over a period of time. That is the process of evolution. So anything in the environment could be triggering mutations, and naturally that process happens over time. In plants, that leads to the evolution of newer crop varieties, newer species. So with irradiation, what we do is that we hasten that process of genetic changes in the seed, and then that seed that is germinating would have an elevated or improved genetic potential, for which we then select our traits of interest. And of course, the seed that carries the genetic change that has been irradiated, and the plant is not radioactive. So it is then taken over multiple generations to select for the characteristic that we are looking for. You mentioned climate change. That is an important characteristic that we continuously select for. And how does climate change impact plants? Normally, when we talk about climate change, we talk about the actual change, the warming global temperatures, the weather phenomena that happens in the world, such as floods, droughts, all of that. But there's also a big impact of climate change on food security, because the plant's ability to perform under rising temperatures is compromised, because heat then affects the viability of pollen grains that fertilizes the ovule to give rise to the seeds or the grains that we eat. So grains, fruits, vegetables, all of these are affected by heat. In addition, if drought happens and there is no moisture in the soil, then the crop is devastated. You cannot get the yield that the farmer is expecting. So that also requires genetic improvement in the crop. Flooding is another thing that has recently found a genetic change that could enable some of the crops to tolerate submerged conditions. So these are the various aspects of climate change that come into play and that affects farmers' income. And that is more and more important for the food security of the developing world. In addition to what I mentioned, high heat temperatures and all that, and drought, there is also warming temperatures that cause diseases and pests to move further north and towards the higher altitudes. And we see that in some of the major pests and diseases that are happening now. In the case of coffee, there is coffee rust that is affecting coffee plantations worldwide. There is the banana fusarium wilt that has been in Asia for a while, and then it is only in 2013 that it started moving to Africa, and then now it has been reported in Latin America. These are diseases like this impacts the farmer and the livelihoods of all of the people who are involved in that chain significantly. So genetic improvement is a major way of tackling these effects of climate change.
All
and
right.
we also breed for improved yields. Yeah.
Shoba, let's go quick so that we all get to other questions too. But one thing, you've convinced me that the plant is not radioactive. You've convinced me that your work contributes to climate change tackling. But how do I accept that you are playing with the genes? What is the genetical modification there? I don't like genetically modified food, so what do I do, for example?
So this is not genetically modified. The genetically modified organisms that we talk about in public conversations is transgenic technology. And more recently, it is also gene editing. And some countries consider gene editing not to be regulated, but most do not. They consider them as genetically modified. But
what does the transgenic mean? Can you just get to that?
What is that? Gene editing?
Transgenic.
Transgenic is when you take a gene from a different organism and put it in a new organism to get that a new trait. For example, Bt-tolerant maize, where you take the Bacillus thuringiensis gene from the bacteria and put it in maize in order to make that maize resistant to insect attacks. So that's transgenic. But the radiation or mutation breeding technology has been used for more than 70 years. And a lot of our crops that we consume are modified in one form or other because of the evolutionary process of mutation, not by genetic transgenic mechanism. Even the Green Revolution earlier on in the 1960s, when famines were hitting, and we were trying to find improved rice and wheat, that came from spontaneous mutations in rice and wheat. And so this is the technology that we're using. So in place of spontaneous mutations, we give irradiation so that those mutations become rapid and we can select from a larger pool.
All right, good. Norman, over to you. We just received a question here. Thank you very much for your question from Stanford Pinto. My question is about how safe and edible are the radiation mutated plants for human consumption for being resilient to climate change? So do I get anything radiation left over from these plants when I consume them?
No, so this is as Shobha just explained. The radiation will simply induce mutations as other factors in the environment will do. And this is physical DNA damage that the plant hurries to repair and occasionally makes the one or the other mistake. So there's simple, small changes in the genome that the radiation causes, which are not any different from changes that environmental influence over the years would cause. So there is no concern whatsoever of the safety of this technology and the food we produce. It's simply so that, so in nature, imagine a large field of grass where you have billions of plants where the evolutionary process happens at a rate of 1 mutation per generation. We can never look at so many plants to select our crops. So we are condensing this process into a number of plants we can actually handle and screen through. And for this, we increase the rate of mutation using the radiation, which is just increasing the rate, but not changing the nature of those changes.
Okay, thank you. Now, Norman, how do you get to choose your crops? You know, Mark and Chantel are fans of tsetse flies. Are you a fan of sorghum or banana? Do you have a particular, how do you get to choose your plants? And who gets to tell you, okay, you now work on this plant?
So thank you for this question. It's important to note that we are the member states' laboratories. We don't do anything without a mandate by our member states. In this particular case, the IAEA has a so-called technical cooperation mechanism, and member states can apply to the IAEA and us for projects, and they already include the crop and the trait they favor. So from those projects, we then select, or actually we implement almost all of these, and strictly work on whatever the member state, and together with the member state, whatever their demand was. From those, I mean, since we're getting so many applications for projects, obviously those TC projects, we have a pretty good idea of what the problems around the world are. And if we recognize patterns, if we see several countries from a region applying with similar crop and issue, then we are free to launch bigger projects focused on R&D, so-called coordinated research projects, where we then gathered teams around the world, specialists on that topic. And within 5 years, we come up with solutions that we then hopefully successfully can deploy through those technical cooperation mechanisms. So in summary, we do work only by member state mandate through direct interactions and applications by member states. And this is how I get to choose my crops. I mean, you see sorghum behind me, which is a photo from last Friday from our field. We obviously have the limitation that in Vienna we can't grow any crop outside, but we have succeeded for most of those. And we don't necessarily have to grow them here because we have a network all over the globe with people where we can grow the crops if we need to.
All right. Before we switch to the insects, can I hear some very concrete crops that have been released for further cultivation that are showing totally new characteristics that didn't exist before? Sorghum is one you are standing behind.
There is obviously, Shoba mentioned earlier the Green Revolution genes, right? The Green Revolution was simple. The start of the Green Revolution came about by a simple trait, which was semi-dwarfism. You can imagine that a crop that you start fertilizing will grow and grow and grow and then eventually become too tall and fall over. So the key of the Green Revolution in the 60s was to have crops that actually stayed small, and so wind and rain couldn't damage them too strongly. And that's obviously a trait that we've been transferring through mutation breeding into other crops, for example, sorghum. Then disease resistance. You have to imagine that a disease and the resistance to a disease or the susceptibility is a communication between the pathogen and the plant, and disturbing that communication through mutations is obviously an easy target for us. And obviously, last week we had a big celebration of a new cotton variety that's now been released a couple of years ago in Pakistan.
Cotton.
Yes, cotton, and that is very, very successful. Mostly, so we are helping the member states to release the crop, so to produce the crop, but then ultimately the release is in the member state by the normal crop release mechanisms. You have to prove that your crop is better than what existed previously, and that's what these member states do by themselves. So we are just assisting in this process.
All right, thank you.
If I may, Ihan, so there are more than 3,000 varieties released from mutation breeding that are cultivated.
Give me the number again, please.
3,365 that is recorded in the database, but this is not the entire.
New varieties of crops, over 3,000.
Mutant varieties that are cultivated all through the globe. And these include a large number of food crops, including rice, that is released through mutation breeding. The largest mutation breeding varieties that are across the globe is from China. And they use a variety of mutation breeding technologies, including radiation by gamma rays, but also space breeding, where they send seeds to the space by space satellite and use that seed from selection. And the second largest variety of wheat that is cultivated in China now is from space breeding. The next country is Japan, that has a lot of mutant varieties cultivated. And then the third is India, but it's all over the globe. And those in the US and in Texas would know the star ruby, the grapefruit, that is one of the grapefruit varieties that have been released a long time back from mutation breeding. So there are varieties across the globe.
All right, thank you. We will probably get back to you with questions. Now let's switch to our other friends, the insects, the tsetse and others. Now, Mark, we've seen the video. That was really quite interesting. And you said there, what I picked there was that the sterile insect technique using radiation to sterilize the male flies was environmentally friendly. How is that? Why is that?
Because, it is environment friendly because the only thing that you're doing is releasing male insects into a target area that cannot propagate. The only thing that they can do is, seek out the virgin females. They mate, and then, after a while, they will then eventually die. Don't forget that they are irradiated, so their lifespan is not that long. So you're actually not infringing upon anything, in nature or the environment.
I'm trying to understand. What do you have against the males? Why not the females? Why do you only sterilize the males?
Well, if you think about it, basically you only need the males. They have the advantage that they can mate, several times. So they have the opportunity to fertilize several or inseminate several wild females. Whereas if you're releasing, sterile females, what can they do? Very often they will mate with the sterile males if they are released together. So actually, it's a waste of sterile sperm. And even if the sterile females is mating with a wild male, there is no impact. So the only impact that you see is when the males are being sterilized, released, and they can then mate with the virgin wild females, making those wild females sterile.
I see. And what animals do you choose? Can you use this on any insect you would wish to?
No, certainly not. And we get many requests from people in member states stating that they have a problem with this insect or that insect. But you have to realize that the phase of the insect that you are releasing should not add to the damage. We have, for instance, fruit flies here in the lab, and it is the larvae of the fruit flies that is causing the damage to the fruit. We are releasing the adult males, and these adult males, they are harmless to the crop. So it's very important to realize that the insect that you are releasing, should not contribute to the problem. Another example is, for instance, mosquitoes. It's the male mosquitoes that we are releasing, and male mosquitoes are not biting the people. They are not transmitting the disease. It's only the females. So if you're only releasing the males, you do not contribute to the problem. So that is a very important principle if you would like to use the sterile insect technique.
I see. And why is this method anyway better than the conventional methods of fighting the insect pests?
Well, as I said in the video, I don't necessarily think that the technology is better, but it is very environment friendly. If we see that the most common way of dealing with insect pests is by spraying insecticides, and this is very bad to the environment. So the big advantage of the sterile insect technique is that it is environment friendly, it is neutral to the environment, and it does not cause any harm.
I see. Thank you. Chantal, I have a question. You come from Africa, South Africa, but the continent where the tsetse fly is one of the main issues for cattle breeding and livelihood. What is the connection of this sterile insect technique really to today's theme, World Food Forum? How do we get this? Is it just the cattle, or is it others as well?
So the tsetse are vectors of the disease. So this actually means that a cattle will get sick with the disease, which is called Ngana for cattle. It's a blood parasite, so it will be inside the blood of the cattle. The tsetse will then come, it will feed from the blood of the cattle, and then it will become infected itself with this parasite, and then it will be able to transmit it to another cattle. So the tsetse acts as the vector that transmits the disease from one individual to the next, to the next, to the next. So when a cattle is infected with Ngana, it is sick and it cannot pick up weight. It cannot produce a lot of milk. It has problems with producing offspring, but it's also it can't be used for plowing your field. So in Africa, many of these farmers are subsistence farmers, and they don't use tractors or anything like that. They use the animals to work their field. And if the animal is sick with Ngana, it's difficult for the animal to be used for working their field. So the disease does not only impact the meat production or the milk production, but it also can impact the farmer's ability to work their field, to plant things like maize and vegetables and other things like this.
I see. One technical question to you, Chantal. Are those lovely insects we've seen with the dots on their back, are they radioactive?
No, they're not. So when we put them inside the radiator and we radiate them out, the dose that we give them is not sufficiently enough to keep to make them radioactive, and they're not. We are just also inducing a mutation inside them, inside the sperm of the males, to make them sterile. So even if we release the males from in the field, they are not very active and they cannot. You have not changed anything inside the male except their ability to have active sperm or sperm that is fertile.
Very quickly, can we hear a few concrete examples of where sterile insect technique is currently used? Against which animals? Against which insects?
So currently we are using it for tsetse flies. They are currently used in Senegal to control their diseases in Senegal. For fruit flies, there is a large facility in Guatemala that produce fruit flies, and this is used in many countries around the world. We also use them in mosquito control for a wide range of diseases like malaria. So Mark, if I don't know if you want to add anything here.
Well, don't forget there are a few big programs against moths. You know, there is a program in Canada against the codling moth that has been going on for more than 25 years. And that has resulted in a valley that is basically insecticide free. And we also have a very nice program against the false codling moth in South Africa. It's actually a privatized company, you know, a private company who is implementing this program, and it's working really well.
Right. Margarita was asking if we can use the sterile insect technique against any insect.
Well, as I said, you know, you can only use it against those insects whereby the phase that you are releasing is not contributing to the damage. We have been asked on several occasions to, you know, develop the technology for desert locusts, for instance. And you know, the desert locust, it eats the crop. So can you imagine what would happen if, you know, with the hundreds of millions that are out there, you add a few hundreds of millions, you know, you would, you know, make the problem worse. So it is only suitable for those insects whereby the phase that you are releasing is not, the development phase that you're releasing is not contributing to the problem.
Right. Just to add to Mark's comment, from a technical side of point of view, you should also be able to know the life cycle of the insect very well. So these insects need to be able to be reared. So you need to bring them inside to the laboratory. You will need to be able to feed the adults and let the adults mate and let the adults be able to produce. So not just release it, that the offspring or the organism that you are releasing doesn't damage your crop, but also that you can technically rear them in large quantities in the laboratory.
Yes, and in most cases, you would need an artificial diet, you know, to produce them. And for many insects, these artificial diets have not been developed yet. So this is, as Chantal points out, this is a very significant issue.
Thank you. Just before we move on back to the plant breeding, Nicola asks, can we use the SIT also for the New World screwworm endemic in South America?
Well, the New World screwworm is, you know, the cell insect technique was actually developed, you know, first for the New World screwworm. You know, it was a problem in the US in the beginning of the last century. Every summer, you saw that screwworm was invading the southern part of the US, and it was moving up north. And, you know, basically they could not keep the insect under control until, you know, this brilliant entomologist came up with this idea. Why not, you know, not to try to kill the insect, but let's see if we can introduce sterility in the population and see, you know, how we can then control it. And, you know, it worked. They only had to wait until the 1950s to find a way to sterilize the insect using X-rays or gamma rays. And, you know, the New World screwworm was eradicated from the United States. It was eradicated from the whole of Central America up down to Panama. And this huge area has been screwworm free, you know, for the last 20 years.
Thank you. Sorry, one more question came in that I also would love to ask this. Chantal, how did you mark those flies with those different colors? We've seen it in the video. I just didn't catch that one.
There is two ways that you can do it. I just painted a little dot of paint on the fly. So if you make it cool in the room, they stand still, and you can just dot a little piece of paint on the thorax, and you can mark them like this. Or of course you can, when they are in the pupa phase, you can put dust around the pupa, and when they emerge out of the pupa shell, they will also get coated with this color dust. And then you can mark them in this way as well. And this is how we...
So that's only for the experimental environment. It's not for a real field application.
No, for field application, you will use the dust over the shells. And this is what they do. When every time sterile males are released in the field, they are colored so that we know which ones were the sterile compared to the field flies. And when we collect individuals back, we can see how many of the individuals that we collected were from the sterile flies that we've released, or they were from the wild individuals in the field.
Thank you. Now, we have 15 minutes left. I'm going to go back to Shoba. We have a question from Esther who says, the technique, the radiation that you use, how does it affect the quality, the nutritional value, and the decomposition process of the crops?
So irradiation and mutation breeding can be used for any trait, including improving quality. The process of irradiation of the seed itself is not going to be affecting the quality of the produce that comes from the crop eventually. So if that is the that is behind the question, then it does not affect the quality in that way. The radiation produces, the radiation is on the seed, and then the seed grows out to a plant, and that plant goes through multiple generations to give the seed that then goes to the farmer for cultivation. There is nothing that affects the quality in that manner. However, just like we affect or improve the ability of the plant to increase yields or produce better iron and zinc in the grain, all of that can be improved through irradiation and mutation breeding by affecting the genetic change in the plant.
And we've seen, I remember from my reading some examples of a new barley variety that grows high up in the Peruvian Andes in 3,500 meters, where it would normally not grow. Is that right?
Yes, yes. The barley mutation breeding program has been very significant in Peru at the UNALM, and they have bred varieties. Normally in the highlands of the Andes, it is very adverse conditions for crop cultivation. So very few crops survive there. And these crops are already attuned to the harsh environments, for example, barley, quinoa, amaranth, all of those. But to make them better growing under these conditions, mutation breeding can be used. And the Centenario variety of barley has been bred to be much significantly improved than the existing varieties. And I think it covers about 17% of the barley area that is cultivated in Peru. That is the total barley area in the country.
That's a huge amount. Thank you for that. Now we have a question on, you mentioned earlier space breeding. How is that working? What is the success rate, and is it more advantageous than the gamma breeding?
That at present...
I'm sorry, before you go, what is space breeding? What are we doing here?
Okay, space breeding is using cosmic radiation to create the mutations. So the further that we are out in space, the higher the cosmic radiation. And so that is similar to gamma radiation, but we are sending the seed to the space. Now at this point, the research is very, very minimal. So we cannot really say how the comparison happens between space breeding and gamma breeding, except in China, but they have not done in-depth investigations. But China is the single country that has been using space irradiation for developing varieties. And since over the last 1.5 decades, they have more than 70 varieties that have been released just from space breeding. And they had a satellite that went out to space in 2006 that was completely dedicated for plants. And 2,000 sets of different varieties of seeds were in that satellite. So they're using that material to breed for newer crops. So that is what's happening in China. Now, at this point, we are at the IAEA, at the Joint FAO IAEA Centre, we are planning to explore space breeding by just starting a feasibility study to send some seeds to the International Space Station. And that is part of a new coordinated research project that will start next year, early next year. In addition to actually sending seeds to space, China also has simulated cosmic radiators. And again, I'm not fully sure how they work, but there is a new facility called the Star Labs that has established in the UAE, which is also looking for simulating space radiation. The interest more recently in space breeding has been extreme when recently there were about 300 cuttings of grapevine that was sent by the French scientists to the ISS, to the International Space Station. And they were brought back down very, very recently, and they're looking at the genetic changes of these grapevines. And again, the main focus is to be identifying material, genetic material that would survive harsh, adverse conditions of growth.
Okay, we don't have much time left. I'm going to come back to all of you about your careers, but I want to ask a question to Mark. Is the SIT, sterilized through radiation? Those insects, are they stressed? Do they show any strange behavior? Does it affect their lifestyle, lifeline when they're released back to the nature?
Well, yes, of course, you have to realize that, we do a lot of nasty things to these insects. We put them, in large volumes, in small cages. We irradiate them with a very, very high dose of irradiation. The irradiation for tsetse revolves around 100, 120 gray. If I give you that radiation dose, you will be dead in 2 weeks from now. So obviously, we throw these insects, into the target area, usually from a plane. So obviously, the quality of the insect is reduced if you compare it to the quality of the wild insect. And therefore, the idea is to outnumber, the sterile ones compared to the wild ones. And we are talking of ratios 10 to 1 up to 100 to 1. 100 steriles for one wild, so that the probability of mating with a wild female is higher for the steriles than it is for the wild ones. So yes, there is a quality reducing element that has to be taken into account. And this is what you have seen in the video, that we are using these cages, these field cages, to simulate a natural environment and to see whether the behavior of the insect has not changed by the things that we have done to them.
Thank you. Thank you. Now, this topic is amazing, both of your areas, but we have 7 minutes left at this World Food Forum live chat on nuclear science for world food programs. Now, I want to get back to a very generic question. How did you get here where you are? And what would you tell our young audience? Because this is a cross-sectional, cross-cultural environment that you work on. You work with statisticians, you work with computer scientists, there is artificial intelligence involved, there is plant breeding, molecular biology in both animals and in plants. But if I'm interested, how do I get to where you are? Let's start with Norman.
I'm pretty excited to share. So I think I was constantly looking throughout my school time and undergrad. I was constantly looking for a place to contribute. It's not like the environmental issues and social issues of this world are new. Back in the 90s, that was also, the young people were outraged by the slow progress we were making. So I was then choosing a career in natural sciences, and I think biology was for me the one to go. And then from there, I did an academic career, a normal one, and ended, well, I'm now here since 2018 at the IAEA FAO Joint Centre, and finally found a place where I can really contribute to the betterment of the world. And if I want to give some advice, just do what you love and think for where you can make your contribution.
Great, thank you. Shobha?
Well, I was born in India, and India is an agricultural country, and I've seen the struggles of farmers, and that is one area that I wanted to work on. And my career has taken me through high-end science in the industry, and also the international agriculture in subsistence farming conditions. And combined with traditional plant breeding and the newer high-end technologies, it's an exciting place and time at this point to contribute significantly to world's food security, and that keeps me going.
Thank you. In between, I can just say at the IAEA, we've mapped out the Sustainable Development Goals, and we are proud to say that we actually directly contribute to 9 out of the 17 Sustainable Development Goals, if not indirectly to all the others. And so the work you're doing in both those labs contribute directly to several of them. So, Mark, what brought you here, and what would you tell our young audience how to get there?
Well, it's in my case, you know, when I was young, I was fascinated by two things. I was fascinated by insects, entomology, and I was fascinated by Africa. It had a real attractive power on me. And then if you put one and one together, you know, you very, very quickly come to tsetse flies, because they are, you know, the root cause of, or one of the root causes of hunger and poverty in Africa. And when I got the opportunity, you know, 30 years ago to come to Zuidersdorf and start as a young, you know, JPO in the lab here, you know, it was an opportunity that I grabbed, and I worked very, very hard. And you know, this is probably one of the reasons why I am where I am. My advice to young people is similar to what Norman is saying. You know, follow your gut feeling and work very, very hard.
Thank you. Chantel?
I think the most important thing in life is to find the place where you fit. So find the thing that fascinates you and that gives you wonder, and see how you can take that thing that you find wonderful and see how you can use it to make a difference in this world. The same with me, and I have the same message than everybody else in this panel. As a young person, I was fascinated by insects, and this is the place that I found that I could work. That doesn't feel like work. I'm playing with insects every day, and this is very fascinating to me. And then when I was young, before I started my PhD, I got exposed to the IAEA, and they really assisted me in taking this passion that I have and developing in such a way that it can be useful and that I can use it in the environment in Africa where I was based. So also my advice is to find the thing that you are passionate about and work really hard to use this to make a difference in the world.
Thank you. Now, to wrap up, it's been wonderful. Thank you for this live chat. Allow me to tell our audience that we're not just the entomologists you see on screen and the plant breeders, the molecular biologists you see on screen. We are a truly cross-sectional professional bunch of people that we include computer scientists, we include radiation specialists, we have very different diverse walks of life here. And we offer very interesting internship programs. Mark mentioned an acronym called JPO. It's a Junior Professional Officer. Basically, the countries send their young, promising people as future leaders to come and join us. We have fellows in the laboratories that our colleagues work. We've proudly trained and benefited from, and they benefit from us, thousands of fellows so far from developing countries that have stayed a few months to a year or longer even at our Zuidersdorf research laboratories. So all of these background and what can you do and how can you get involved with us, you can find on our website, iaea.org, or even the Joint Center, the FAO IAEA Joint Center for Food and Agriculture. Thank you very much, all of you, Norman, Shoba, Mark, Chantel, our audience. We appreciate that you've dedicated your 1 hour to us today. We will reach the Sustainable Development Goals together and with nuclear science helping you. Thank you and greetings from Vienna.
Thank you.
Thank you. Bye-bye.
Bye.